Hello my dear students, a very good afternoon to all. I welcome you all to the most interactive and interesting live session. This is Vasavi Ma'am from Neet World, your biology educator for Foundation and CBSE. Yes, students, in the last class we discussed about the plasma membrane structure as well as its transport mechanisms from the chapter Cell, the Fundamental Unit of Life from the 9th CBSE syllabus, right children? Yes. So, coming towards the today's particular session, so from today we are going to start all the cell organelles children, okay. We are going to study each and every cell organelles in an order wise, yes. So, in today's particular session, we are going to discuss about the cell organelle that is called as nucleus, right, yes. So, today we are going to discuss about the nucleus, yes. Yes. So, children, the nucleus is a double membrane bound cell organelle, okay. So, in the eukaryotic cell structure, we are going to have the double membrane bound cell organelles, single membrane bound cell organelles as well as the non-membrane bound cell organelles, okay. So, the nucleus is a cell organelle which is a double membrane bound, okay. It is bound by two membranes, okay. And if you look into this nucleus, generally we call the nucleus as, there is one interesting fact about some cell organelles. So, apart from that, for the nucleus we are going to call it as brain of the cell. Nucleus is called as brain of cell, okay. Yes. So, why we are calling the nucleus as the brain of the cell, okay. So, you are going to understand it in the upcoming slide children, yes. The nucleus is called as the brain of the cell. Why means it is going to possess some important characteristics, like it is going to store the genetic material. That is the reason, okay, that is the reason we are going to call the nucleus as the brain of the cell, as well as the nucleus is involved in controlling all the cellular activities which are required for the cell's metabolic activity children, yes. So, now, yes, so actually how the nucleus is going to look like, okay. The nucleus is a spherical shaped body. The nucleus is a spherical shaped structure which we can find in the cytoplasm. Inside the cytoplasm there is a spherical shaped structure called as the nucleus, okay. This is what all you know, okay. But apart from that, there is an extra information here. So, inside the cell, imagine this as a cell, and inside the cytoplasm, yes. So, inside this cytoplasm, yes, this is a cytoplasm, whatever I am marking it with the red color, yes. Inside this cytoplasm we have the nucleus, okay, yes. We have the nucleus which is going to contain the genetic material. This is what all you have know and all you have read in the previous sessions, okay. In the previous classes, you all know that the nucleus is one spherical shaped, it is a round bound membrane bound structure which is going to present in the cytoplasm of the cell. But that is not true, because the nucleus will not always be in a spherical shaped structure, okay. The nucleus structure is not always in circular manner, okay, but the nucleus is going to vary. The shape of the nucleus is going to vary in different tissues and different organisms. In the different microorganisms and in some different tissues, the shape of the nucleus is going to vary. What we have said here, the nucleus is a spherical shaped body, but here if you see these given pictures here, the nucleus is going to vary its shape. So, how it is going to vary, how the shape of the nucleus is going to be present in each and every tissue and in some microorganisms, we are going to look into that now, yes. Yes, if you see this picture, it is called as paramecium. What is paramecium children? It is a paramecium, okay. So, the paramecium is a protozoan, okay. The paramecium is a protozoan which is going to contain two nucleus in it. That is called as the binucleated microorganism, and this protozoan, the paramecium is going to contain only single cell, hence it is called as protozoan, and it is a unicellular microorganism. In this paramecium, we have the binucleated structure, Okay, can you see this purple colored structure? Yes. So it is going to contain two types of nucleus. One is macronucleus and the smaller one is called as the micronucleus. Okay. So actually the paramecium structure, the paramecium microorganism is a binucleated microorganism which is going to contain the macronucleus as well as the micronucleus. The micronucleus is very small when compared to the macronucleus. Okay. And yes, and next this is the picture. Okay, the given picture, it is called as the skeletal muscle. It is a skeletal muscle. You will learn about these muscle structures in the next upcoming chapter, that is you are going to learn it in the tissues chapter, children. Yes, it is a skeletal muscle. Here if you see, the skeletal muscle is a multinucleated muscle. The skeletal muscle is a multinucleated, it means it is going to contain more than one nuclei. Can you see here? Yes, let me choose another color, by that it becomes easy to identify. Yes, it is a multinucleated tissue. Which one? The skeletal muscle. And next, coming towards the WBCs, white blood cells. In the white blood cells, if you see the structure of the nucleus, it is lobed in structure. Can you see here? Instead of presenting it instead of giving the circular shaped appearance, here the nucleus is going to form lobed like structure, children. Yes, this is a lobed nucleus which you can observe in the white blood cells. See here, 3 lobes are going to form, 1, 2 and 3. Hence it is called as trilobed, bilobed and monolobed. Okay, these are the lobed structures of the nucleus which we can observe in the white blood cells, WBC. Yes. And next, coming towards this plant cell, about the plant cell and animal cell also I am going to give you a crystal clear idea in the next upcoming session, children. So as if now just look into this diagram. So in this plant cell there is a nucleus. In the plant cell also nucleus is going to be present, but due to one reason, okay, due to the presence of the vacuole. This is a vacuole which is going to be present in both the plants and animal cells. But when it comes to the plant cell, in the plant cell the vacuole is going to occupy the major portion when compared to the nucleus. Can you see? Yes, this is a vacuole which is going to contain the fluid filled material called as sap. The vacuole is also a cell organelle. It is a single membrane bound cell organelle which is going to contain the sap. Sap is nothing but which contains some fluid filled material along with some minerals and nutrients, children. Yes, and here if you see, this is the nucleus. So now what this vacuole is going to do, it is going to occupy the major portion in the plant cell. So due to this reason, the cytoplasm and the nucleus is going to be pushed towards the periphery. The nucleus as well as the cytoplasm is going to be placed towards the end of the plant cell, and this vacuole will occupy the maximum portion in the plant cell. Okay, so the nucleus is not in the center, but it is going to be present towards the periphery in the plant cell. Yes, and next if you see the animal cell, in the animal cell the nucleus is present in the center of the cell. This is a nucleus which is going to be present in the center of the cells in the animal cells. And next here comes a simple squamous epithelial tissue. About the tissues you are going to learn in the next chapter, children. Okay, that is also very interesting chapter. Yes, after completing this we are going to move to the next chapter, that is the tissues. Okay, there you are going to learn about the animal tissues. Okay, now just I am giving you a small introduction about it. Yes, this is the simple squamous epithelial tissue. Okay, so in this simple squamous epithelial tissue you can find the circular nucleus. Okay, centrally located in nucleus and flattened nucleus is going to be present. Okay, the nucleus is present at the center, but the shape of the nucleus is somewhat flattened like this. Okay, it is present in the simple squamous epithelial tissue. So this is about the structure of the nucleus and its shape. Okay, so if you see here, the shape is going to vary in different cells, different tissues and different microorganisms. In the paramecium, it is a binucleated which is going to contain some enlarged nucleus and one micronucleus. And next, if you observe the nucleus in the skeletal muscle, instead of going to be present only one, it is going to contain many nuclei, hence it is called as the multinucleated skeletal muscle structure. And next, these are the white blood cells where you can observe the lobed structures of the nucleus, and it is a simple squamous epithelial tissue. Here the nucleus is present. Okay, each and every cell is going to contain the nuclei, but those are some flattened structures and those are centrally located. And here it is an animal cell where the nucleus is located in the center of the cytoplasm, and it is a plant cell where the nucleus is going to be present towards the periphery due to the presence of vacuole. The vacuole is also a cell organelle which is going to contain the sap. Sap is nothing but which is made up of both some fluid filled material as well as along with that you are going to that is going to contain some minerals and nutrients. Okay, about this also we are going to learn in detail in the further sessions. Okay, this is about the general introduction and some extra information regarding the nucleus. This is also going to help you in the higher grades children. Yes, now we are going to discuss about the structure of the nucleus, the diagrammatic view of the nucleus along with its functions in detail. Yes. Yes, children, look here. This is the structure of the nucleus. Okay, the nucleus is going to be called as true nucleus. True nucleus. We are calling it as the true nucleus, and we can find this true nucleus structure only in the eukaryotic cells. Eukaryotic cells. Okay, why we are calling this particular nucleus as a true nucleus in the eukaryotic cells? Because due to the presence of some important characteristic features, those are presence of nuclear envelope and next presence of the nucleolus. We are going to discuss about each and every term, each and every part in detail. Okay, this is just an outlook children. Yes, and next nucleolus, and next one is nucleoplasm, and next chromatin material. Okay, the presence of all these four structures, those are nuclear envelope, nucleolus, and the next nucleoplasm, and next chromatin material. These four characteristics are going to show the major differences in between the prokaryotic and eukaryotic cells. So, once quickly I will recall you how the prokaryotic cell is going to be vary from the eukaryotic cells. In the prokaryotic cell, we have observed that there is a circular genetic material which floats freely in the cytoplasm of the cell. True? Yes. This is called as the primitive type of nucleus which we have observed in the prokaryotic cells. Okay, but when it comes to the eukaryotic cell, there is a genetic material which is going to be surrounded with the nuclear envelope. Okay, and along with this nuclear envelope, that is going to contain few more characteristics. Clearly, you can observe here. Okay, now we will discuss about the nuclear envelope, nucleolus, then next nucleoplasm and chromatin material. Yes. Just a minute, yes. So, based from here also you may get the case study questions children. Okay, from here we are going to learn all the cell organelles based on the cell organelles, based on the arrangement, based on its location also you are going to get the case study questions. We are going to solve it in the next, after completing all the cell organelles, we are again going to solve the case study questions with respective to the particular cell organelle. Yes. So, nuclear envelope, what it is? Nuclear envelope. We have called the nucleus as a double membrane bound cell organelle, right? So, the nucleus is going to contain two membranes. The nuclear envelope is going to contain actually the nucleus is made up of two membranes. It is going to be covered entirely with two membranes. So, one will be the outer membrane and next inner membrane. Clear? Okay, outer membrane and then next inner membrane. So, in between these two, in between the outer membrane and inner membrane, there is a fluid filled space that is called as perinuclear space. If you can observe here, this is called as the outer membrane and inner membrane. In between these we have perinuclear space. Just I am writing it here. Yes, in between these two we have a space filled with the structure called as perinuclear space. Yes, it is called as the perinuclear space, and yes, this is about the nuclear envelope. The outer membrane and inner membrane along with the perinuclear space is called as the nuclear envelope. Yes, and next coming towards the nucleolus. Okay. The nucleolus is a spherical shaped body. The nucleolus is a spherical shaped body. It is a non-membrane bound in structure. Okay, it is non-membranous. There is a no membrane which covers the nucleolus. Okay, if you see this diagram here, you can see this is the nucleolus, which is a spherical shaped body and which is stained in nature. Okay, that is a colored stained spherical body which is present inside the nucleoplasm. See here, this is the nucleolus. It is a spherical shaped stained body which is present inside the nucleoplasm. Yes, and next if you see here, this is called as the nucleoplasm. This is nucleolus, spherical shaped body, and here the nucleolus is present in the semi-fluid filled substance. Here if you can observe, yes, this all white colored matter inside this inside which the nucleolus is located, that is called as the nucleoplasm. Okay, and this nucleoplasm is going to contain few important enzymes. The nucleoplasm will contain the two enzymes, those are called as DNA polymerases and RNA polymerases. DNA polymerases and RNA polymerases are going to be present inside the nucleoplasm. Okay, and these DNA and RNA are called as nucleic acids. These are called as the nucleic acids. Okay, and DNA is called as deoxyribonucleic acid and RNA is called as ribonucleic acids. This DNA polymerase is involved in making the DNA and RNA polymerase is involved in making the structure of RNA. Clear with the concepts? The DNA polymerases and RNA polymerases are going to be present inside the nucleoplasm. These are actually the enzymes which are going to be involved in making the DNA and RNA, deoxyribonucleic acid as well as the ribonucleic acid. Yes. Now, we are going to discuss about the chromatin material which is present inside the nucleus. Yes, I will erase each and everything. By that you will get the clear idea where actually the chromatin material is going to be present. Yes. So, look here children, inside this inside the nucleoplasm, if you observe, here are some thread like colored structures which is going to be present entirely inside the nucleoplasm. Can you see this, whatever I am drawing? Yes, this is called as chromatin. Clear? Chromatin. So, chromatin material contains chromatin material. So, first observe the term. Okay, first try to understand the term what actually the chromatin material means. So, the chromatin material, chroma in the sense color. Okay, chroma means color and tin in the sense thread like structures. Okay, yes, chromatin material in the sense the color-filled network like thread like structures which are going to be present inside the nucleoplasm. Clear with the term chromatin material in the sense the color-filled thread like structures. The color-filled thread like structures which are present inside the nucleoplasm. Okay. So, actually the chromatin materials is going to look like this. If suppose if it is a nucleus, inside this we have the chromatin material, inside the nucleus only we have the chromatin material. Now, if you see here, for example, if this is a nucleus and inside this we have the nucleoplasm, and the next here we have the chromatin material, which is colorful network like thread like structures which is present inside the nucleoplasm. So, now when you are going to start separating all those chromatin material, okay, when you start separating those colorful thread like structures, they actually look like in this manner, children. Clear? So, when you start separating them, they are going to appear like single thread like structures. Clear? So, now we are not going to call it as chromatin material anymore. Why means here we cannot observe the network or reticular like structures. Now, we have separated all those chromatins. So, now when you separated these chromatin, they are going to appear individually. Okay. And so, each chromatin during the cell division, it is going to form thickened structures. So, try to understand slowly. So, during the cell division, these chromatin structures actually first they are going to be somewhat thin like structures, but during the cell division process they will give thickened appearance like this. They will enlarge its size in and they are going to become thick in nature. Why this is going to happen means during the cell division, due to the dehydration process, due to the loss of water, the chromatin is going to give thickened appearance, and this chromatin now when it is going to give the thickened appearance during the cell division, it is going to contain the DNA. Suppose if this is the chromatin material. So, inside this chromatin material has become thick now due to the loss of water. Just I am filling it. Yes. So, inside this chromatin material now we have the genetic material that is called as DNA. From here please pay your attention, based on this again you have to study different types of chromosomes. Yes, children. So, this is actually a single chromatin structure. First usually before the cell division they are actually going to give the thin appearance, but during the process of cell division they are going to gain their size, they are going to become thickened structures due to the dehydration process, that is nothing but due to the loss of water they will give the thickened appearance, and inside this chromatin only we have the DNA. Is it visible? Yes, inside the chromatin only we have the DNA double helical structure. This is all about the chromatin. Understood? Understood? Clear with the chromatin structure? Chromatin is present, chromatin material is present inside the nucleoplasm. That chromatin material, we are calling it as a chromatin material. That is because those are colorful network like or thread like structures which are present inside the nucleoplasm. When we are going to start separating all those chromatin material, they are going to be appeared as single individual thread like structures. But during the cell division process, that is going to acquire the size in thickened shape and it is going to contain the genetic material that is called as the DNA. And during cell division only they will give the rod shaped appearance, that particular rod shaped structure appearance that is called as, yes, this is called as chromosomes. Yes. Can you see here? Yes, during the cell division process only that chromatin material will acquire the rod shaped structures. They will form the rod shaped structures, those are called as the chromosomes. It will be giving the appearance in this form. Okay. These are actually the chromosomes. So, in between this here center, here we have the non-membrane bound transparent spherical body that is called as a centromere. This is called as centromere, which is located at the center, and to this centromere only the chromosomes are attached. Usually we call these two rod shaped structures as P arm and Q arm. Clear? Clear with the concept? This is a centromere. It is a spherical shaped structure which is located in the center, and to the centromere only what is going to be attached? Chromosomes, okay, and that chromosomes are usually we are labeling it as a P arm and Q arm. How we got this rod shaped structures, children? During the cell division, the chromatin is going to acquire thickened structure which is going to contain the DNA and it will give the rod shaped appearance. Those are only called as the chromosomes. Yes. Now, based on this center positioned centromere, okay, based on this centromere we are going to divide the types of chromosomes, okay, that chromosomes are going to be divided into 4 types based on the position of centromere. The centromere may not always be present in the center, but sometimes it is going to vary its position slightly. So, due to that variation only we have classified the chromosomes into 4 types. Those are, this is very very important for the competitive examination purpose like NTSC and Olympiads. For that particular examinations you need to have you need to learn the clear cut information, you need to learn extra regarding the nucleus, okay. This is actually a competition window, okay, it is located in your NCERT textbooks also, but the students who are going to prepare for the competitive national level exams, they have to learn each and every concept in a clear manner. You need to learn extra, okay. Learning extra always gives you a better results when coming towards the particular type of questions. Yes. So, now we are going to learn about the types of chromosomes based on the position of centromere. Yes, based on the position of centromere, these are the types of chromosomes. Yes. First one is the metacentric chromosome. Children, please pay your attention and look into these diagrams, by that it becomes easy to answer, okay. Yes, every answer, the total answer is present only in the diagrams itself, okay. Once if you are perfect with these structures, then you can easily understand the concept. Yes, these are the types of chromosomes we have classified based on the position of centromere, and the first one is the metacentric chromosome. In the metacentric chromosome, there is a centromere which is present in the center, okay. Yes, in the metacentric chromosome we have the centromere which is located in the center, okay. And next, in the submetacentric chromosome, if you observe, the centromere is positioned slightly off the center. Instead of locating it into the center, it is going to be present slightly upwards or downwards, but it is not actually in the center. But in the metacentric chromosome, when the centromere is present at the center, there is going to be present equal arms. We can find the equal arms, equal P and Q arms. We have labeled it as P and Q arms, no? Yes, these are going to be equal in the metacentric chromosome. In the submetacentric chromosome, unequal arms are going to be present. What, children? Unequal arms we can observe in the submetacentric chromosome, that is due to the centromere which is located slightly upwards or slightly downwards. And in the acrocentric chromosome, this is the acrocentric chromosome, and the centromere which is located in the acrocentric chromosome is, it is located slightly towards the one end, okay. The centromere, what you can observe here is, it is located slightly to one end, that may be either towards the top or else it may be towards the down, but it is not going to be present actually in the middle. So, during this particular case, we can observe 2 extremely small arms and 2 extremely long arms. Clear with the concept? By looking the diagram itself, you can understand what actually, how the chromosomes are going to be divided, okay. Yes, this is the centromere, and due to the position of the centromere towards one end, the arms, the P arm and Q arm, those are 2 are extremely small and 2 are extremely long. Yes, next, coming towards the last type of chromosome, that is telocentric chromosome. So, here the centromere positioned towards the terminal end, okay. The centromere is positioned towards the last, okay, and due to this reason we cannot find the two arms, okay. We cannot find both the P arm and Q arm in both the directions, but actually both arms are located only in one side. See, this will be the P arm and Q arm which are located only on one side. If you compare the two arms when coming towards the acrocentric chromosome, we have equal distribution of the arms, that is two arms are present on one side and two arms are present on the next side. But coming towards the telocentric chromosome, there is a centromere which is located in the terminal position. That is the reason we have the arms present only in the one side. Clear with the concept? This is all about the types of chromosomes. Okay, this is about the types of chromosomes which are divided based on the position of the centromere. That is metacentric chromosome where the centromere is at the center and two equal arms. Submetacentric chromosome, centromere is slightly off the center and two unequal arms. And acrocentric chromosome, the centromere is slightly present to one end and it gives two extremely short arms. Two arms will be extremely small and two arms are going to be present extremely long. And next one is the telocentric chromosome. The centromere is located at the terminal end, giving the arms on one side itself. Clear with the concept, children? See here. Then look into the functions of the nucleus. It controls all the cell's metabolism. Okay, the term metabolism in the sense sum of all the chemical reactions which are going to occur in our body of the organism. And next, see here, see this statement, chromatin stores the genetic information required for the development of the organism, the development process like reproduction, metabolism and behavior. Okay, to perform the reproduction, metabolism and behavior, to perform all these activities, we need the genetic material, right? Where the genetic material is actually going to be present? It is going to be present inside the chromatin. Yes. So, here is one important statement regarding to the examination point of view. Yes. So, here is one most important statement which you can get in the 1 mark type of questions. This already you people has learned. The nucleus is discovered by dash. The nucleus is discovered by, in the fill in the blanks you might get such type of questions, children. Already you have discussed about this in the, where you have discussed about this in the discoveries of cell. Okay, in the subtopic discoveries of cell, there you learned all the theories regarding to different types of scientists regarding which is relevant to the cell. So, the nucleus is discovered by Robert Brown in the year 1831. Clear with the concept? Where he discovered that nucleus? Okay, he discovered the nucleus in the roots of orchid plant. Very important. Please try to memorize everyone. Roots of orchid plant. Yes. And next, there is one exception. See, children, exceptions are very important as they are actually going to prove the rule. Okay, there is one exception. What is that exception means? Nucleus is present. Okay, nucleus is present in the organisms. Okay, in our body also nucleus is present actually. So, but the nucleus is absent in RBC. What are RBCs? Red blood cells. Okay, RBCs, these are erythrocytes which are also called as red blood cells. Yes, inside these RBC we cannot find the nucleus. Nucleus is absent in the RBC. This is one exception. Please try to remember. See here. If you are clear with all the concept, you can understand this easily. These are the very most important points which are directly extracted from your NCERT syllabus itself. Yes, nucleus is called as the brain of the cell and it is also called as director of the cell. Why it is called as brain of the cell? Because it stores the genetic information which is required for the development of an organism. And next it is called as director of the cell, as it is going to control the metabolism of the cell. It is called as the director of the cell. And as you already learned, it is going to contain 4 important components: nuclear membrane, nucleoplasm, nucleolus and chromatin material. These are very important. If you are clear with this, you will easily understand how it is going to vary from the prokaryotic cell. This is all about the nucleus, children. Yes. Yes, next coming to words, one more important cell organelle, that is endoplasmic reticulum. This is also very important. It is very short subtopic, you can easily remember it. Yes, Endoplasmic reticulum. First always try to understand the particular term, okay. If you understand this term, you can easily understand how it is going to be present, how the location is going to be present. See here, endo in the sense inside, okay, and plasmic in the sense which is present in the cytoplasm, and reticulum in the sense which is going to present network like structure and which is going to show some compartment like structures in the cytoplasm. Just simply I am writing it as network. So now endoplasmic reticulum in the sense, please try to look here, endoplasmic reticulum, endo in the sense inside, plasmic means inside the cytoplasm, and reticulum means network like structure, clear. So endoplasmic reticulum in the sense inside the cytoplasm there is some compartment like structures, okay. We can observe some compartment like structures or network like structures which are present in the cytoplasm, that is called as endoplasmic reticulum, yes. So from here we are going to learn the endomembrane system. This is also very important. It is just some extra information. The term endomembrane system, I think you do not have the basic idea about it. So please let me make this concept clear for all of you children. Yes, it is called as endomembrane system. Okay, this is the endomembrane system. So if you see the nucleus, to the nucleus external side there are some small granular like structures which are going to be present, okay. Once I will show you the structure of the nucleus, by that it becomes easy to you, yes. So if you see the nucleus, there are some granular like structures which are present towards the exterior towards the outer surface of the nucleus. Those are called as ribosomes. See, can you see all these yellow colored dot like structures? These are called as ribosomes, okay. The nucleus is going to be present rough in structure due to the presence of ribosomes only. Now we are going to learn about the ribosomes, okay, yes. And this complete nucleus is continuous with one cell organelle, that is called as endoplasmic reticulum. Clear with the concept, okay. And this actually I am going to explain you in the next slide, but just once I am showing the structure how it is going to be present, okay. The nucleus is going to be give the rough structure due to the presence of ribosomes on its external surface. And to the nucleus what is going to be continuous? To the nucleus what is attached, children? Endoplasmic reticulum, yes. Yes, see here, this is actually the endoplasmic reticulum and this is the nucleus and this will be the nucleolus. Here we have the nucleoplasm and the next chromatin material. To this outer surface the cytoplasmic, to this outer surface the compartment like structures which are going to be arranged, that is called as endoplasmic reticulum. And endomembrane system in the sense, endomembrane system includes few important cell organelles which are going to communicate with one another, okay. Those all are interlinked. So those are nuclear envelope, yes, this will be the nucleus. To the nucleus what is present? Nuclear envelope, I am writing it as N and E, nuclear envelope. And outside the nuclear envelope there is a cell organelle which is going to be continuous with the nuclear envelope. For the nuclear envelope the endoplasmic reticulum will attach. This is called as ER, endoplasmic reticulum. And towards the endoplasmic reticulum there is one more cell organelle, that is called as Golgi body. About all this we are going to learn now. And in between this we have the vesicles and then next lysosomes. Here it is not given, but lysosomes are also going to be present. And then next plasma membrane, okay. These all are going to be called as, these all together called as endomembrane system. Why we are calling it as endomembrane system? In the sense each and every cell organelle is going to communicate with the another cell organelle. So how actually the process is going to occur, we are going to learn now in detail, yes. See children, this will be the endoplasmic reticulum. So, if you observe the structure of the endoplasmic reticulum, it is going to contain three most important structures. Those are cisternae, vesicles and tubules. Okay, the endoplasmic reticulum is actually going to contain three different structures. Those are called as cisternae. Cisternae are always flattened structures. Okay, and next vesicles. Clear? The vesicles are small circular membrane bound structures. These vesicles will actually carry the proteins. Clear? These are circular in nature. And next one are tubules, which are elongated in structure. If you can observe here, by that you can understand children. These are the tubules and these are cisternaes. Clear? Cisternaes are flattened structures, tubules are elongated structures, and in between them we have the vesicles, which are circular in nature and which are going to carry all the proteins. Yes. So, now the endoplasmic reticulum is actually divided into two types. One is rough endoplasmic reticulum and next one is the smooth endoplasmic reticulum. And smooth endoplasmic reticulum. Yes. See children here. So, we have two types of endoplasmic reticulums. One is rough endoplasmic reticulum and another one is the smooth endoplasmic reticulum. See, this is the rough endoplasmic reticulum, where you can observe the granular like structures called as ribosomes. First, observe these compartment like structures. This is called as the cisternae and these yellow colored dotted structures are called as the ribosomes. Okay. These ribosomes, the presence of ribosomes on the surface of the endoplasmic reticulum will give the structure called as rough endoplasmic reticulum. Why we are calling it as rough endoplasmic reticulum children? It's because we have the ribosomes which are present on the surface. Now, these ribosomes are involved in synthesis of proteins. Ribosomes are involved in synthesis of proteins, right? And next, if you see here, suppose imagine if this is a cell and inside the cell you have the cytoplasm, and inside the cytoplasm the many membrane bound, non-membrane bound cell organelles are going to be exposed freely. Yes. Now, inside the cytoplasm, inside the cell, the whatever the fluid is present towards the outer side of the endoplasmic reticulum is called as extraluminal space. I am writing it as ELS, extraluminal space. That is also, you can also call it as the cytoplasm. And there is a space which is enclosed inside these compartment like structures called as the cisternae. Inside these also there is a fluid filled substance that is called as lumen. Yes. Okay. See here, whatever I am drawing it, yes, first let me make you a clear. See here children, the dark pink colored structures, this color whatever I am drawing now, this is called as lumen. Clear? So, the lumen is present inside those cisternae and extraluminal substance is present towards the outer surface of the endoplasmic reticulum, and we have two types, those are rough and smooth, rough ER and smooth ER. Rough endoplasmic reticulum is involved in synthesis of proteins, and in the smooth endoplasmic reticulum, here in this structure you cannot find any granular like structures, that is called as smooth endoplasmic reticulum. The smooth endoplasmic reticulum is involved in synthesis of lipids. Clear children? About these vesicles and vacuoles you are going to learn it in the next subtopic, that is in the Golgi bodies. Yes, now we will look into the functions of the endoplasmic reticulum. See students, whenever you are going to study all the cell organelles, it is very mandatory to memorize each and every diagram. You have to practice the diagrams well, and you need to learn the functions and you need to learn about its theoretical part. Okay, based on this you are going to get the case study questions as well as some long questions which are going to come for 4 marks as well as 5 marks. For that things to score, you need to learn all the cell organelles in detail along with structure as well as its function. Yes, now next coming towards the functions of the ER. So it is going to form 4 or 5 most important functions. First, it is going to support the cytoplasm by providing the kind of cytoskeleton to maintain the shape of the cell. It means, suppose if this is a cell, the endoplasmic reticulum, okay. Sometimes the endoplasmic reticulum may span the entire plasma membrane, it may be surrounded towards the cell, and sometimes the endoplasmic reticulum may be attached to the nucleus, and sometimes it may be present away the cytoplasm. Clear? So, this endoplasmic reticulum by performing by these particular locations, it is going to give the shape of the cell. Clear? Did you understood? Yes, this is the endoplasmic reticulum. Sometimes it may span towards the entire cell, it may span towards the entire plasma membrane. Sometimes it may be attached to the nucleus, or else sometimes it may be present away from the cytoplasm. So, by this it is going to give a proper shape to the cell, and it provides the extensive site for proteins and lipid synthesis. These two are very important. Which is involved in protein synthesis? Rough ER, and lipid synthesis smooth endoplasmic reticulum. Yes, and next it transfers the proteins from cytoplasm to the nucleus. See, proteins, it is going to form the proteins, okay. Endoplasmic reticulum will form the proteins. So, how it is going to transfer from the ER to the nucleus? So, inside the nucleus, this is one extra information to all of you children. This is actually you are going to learn in the higher grades, but as it is a statement, I am just explaining you. See here. So, for the nucleus, from the external surface there are nuclear pores. See, in this manner, in this shape, the nuclear pores are going to be present towards the outer surface of the nucleus. Yes, to the nucleus, what is going to be attached? ER. Suppose if this is a nucleus. So, to this nucleus, nuclear pores are present. So, this is a rough endoplasmic reticulum where ribosomes are attached, which are involved in synthesis of proteins. So, now these proteins will travel from rough ER, and they are going to be freely suspended towards the inner environment of the nucleus by the involvement of nuclear pores. Clear with the concepts? Nuclear pores are present on the outer surface of the nucleus. So, from that nuclear pores, the proteins will travel. Where the proteins are going to be synthesized? On the rough ER. This is actually the process how proteins will enter into the nucleus. And next, smooth ER is involved in detoxification in the liver of vertebrates. Clear? Inside the liver, it is going to break some toxic substances into the simpler ones inside in the vertebrates. This is also one major important functions which is involved in detoxification of the liver. This you may expect for the MCQ, for the multiple choice of questions you may get this concept children. Detoxification of liver in the vertebrates is going to be occurred by the involvement of cell organelle called as the endoplasmic reticulum. Inside the endoplasmic reticulum, only smooth ER will involve. Yes, clear with the concept children, clear with the nuclear structure, its functions and types of chromosomes. The types of chromosomes are very important. Please try to memorize and try to grasp the concept children. And next, this is all about the endoplasmic reticulum. So, in the upcoming session you all are going to learn about the one more interesting and important cell organelle. So, for that cell organelle we can call that cell organelle as a postman of the cell. So, this is a question to all of you children. So, please try to answer which cell organelle is called as the postman of the cell. So, please stay tuned and please like, share and subscribe to Neet World. Thank you.